Welcome to the Deep Dive. Today, we are opening up the blueprint for what is probably the most critical logistics and security system in all of biology.
The cell membrane.
Exactly, the cell membrane. If you picture the cell as this, complex, self-sufficient factory, the plasma membrane isn't just the wall.
No, not at all. It's the ultimate gatekeeper, constantly balancing the need to import fuel and export waste.
All while fighting a chaotic, unpredictable external environment.
It's an astonishing feat of engineering, really, and it's essential for maintaining that perfect internal stability, what we call homeostasis.
Yeah.
And the core challenge isn't just opening the gates, it's being ruthlessly selective about what moves when and how fast.
Okay, so our sources today lay out this entire logistical operation.
They do. They distinguish between the two fundamental modes of traffic flow: passive transport, which is sort of the free ride,
And active transport.
and active transport, which is the expensive, energy-intensive process that allows cells to defy the usual flow of chemistry.
Okay, let's unpack this. Our mission is to zoom in on the counterintuitive components that give the membrane its stability, understand how molecules ride concentration gradients, and then dive deep into those expensive uphill battles the cell fights using its power currency, ATP.
And we've got some absolutely mind-bending theoretical findings on how the most efficient channels actually work.
So when we look at the membrane itself, the foundation is the phospholipid bilayer.
Right, these are the two layers of lipids, but, if you stop there, you're really missing the complexity.
Because that structure is packed with other components.
It is, most notably proteins and cholesterol. The bilayer provides the basic structure, that hydrophobic barrier that keeps water-soluble things out, but the heavy lifting is really done by the proteins.
And we group them by where they sit.
We do. You have the integral proteins, which are embedded right into the main fatty structure, often spanning the entire membrane.
Which makes them incredibly hard to extract in a lab setting.
Very difficult. And then if those integral proteins are the hard-working gates, the peripheral proteins are, a bit different.
More like dynamic regulator.
Precisely. Peripheral proteins are only loosely associated with the surface, either the inner or outer face, making them much easier to extract. Their role is often regulatory or signaling, unlike the integral proteins doing the hard transport work.
But this is where the structure gets genuinely paradoxical.
With cholesterol.
Yes, with cholesterol.
Yeah.
Most people associate it with rigidity or, bad health. But in the cell membrane, it plays this sophisticated dual function that makes it a biological temperature buffer.
It's absolutely crucial in eukaryotic cells. Think of cholesterol as the molecular cement and thermostat all combined.
Okay.
Its rigid steroid ring system generally reduces the mobility of the fatty acid chains in the phospholipids.
So it makes the bilayer less permeable to small water-soluble ions like sodium or hydrogen.
That's it. It stabilizes the structure, giving the membrane integrity.
So it's reducing permeability, but what about the temperature buffering? How does it manage fluidity bidirectionally?
It's brilliant adaptive engineering. At high temperatures, the membrane might start to melt and become excessively fluid.
Too leaky, too loose.
Exactly. Cholesterol steps in and actually decreases that fluidity by restraining the movement of the phospholipids. It prevents a cellular meltdown.
And at the other extreme, when things get cold, everything tends to stiffen up.
Right. At low temperatures, phospholipids want to cluster together and crystallize, turning the membrane rigid and dysfunctional.
So cholesterol gets in between them.
It intercalates between those chains, acting like molecular spacers. It prevents that tight, rigid packing, ensuring the membrane remains pliable and functional even when the external temperature drops. It's the ultimate stability agent.
So once the fortress has achieved its perfect stability, thanks to that temperature buffering cholesterol, the real challenge begins: managing the continuous flow across that barrier.
And that brings us to the movement that doesn't cost the cell a dime.
Passive transport.
Passive transport. It's simply the cell taking advantage of the concentration gradient. It's coasting downhill. Molecules move spontaneously from an area of high concentration to low concentration until they reach dynamic equilibrium.
No energy required.
None at all. And we have two main avenues for that downhill ride. First, the most straightforward: simple diffusion.
Simple diffusion is the direct passage through the phospholipid bilayer itself.
Yes, and this is reserved only for tiny, non-polar hydrophobic particles. Think oxygen, carbon dioxide, nitrogen. They don't mind the fatty interior of the membrane and just slip right through.
But if you're large, or if you're charged and polar, you hit that fatty layer and you're instantly repelled.
And that's where the assistance comes in the form of facilitated diffusion.
Right. Facilitated diffusion absolutely requires specific transmembrane proteins, either channel proteins, which are like tunnels, or carrier proteins.
Which physically bind and shuttle the molecule across. Things like glucose or amino acids need this assistance. And this mechanism introduces some interesting kinetics.
It's like rush hour traffic, isn't it?
That's a perfect analogy. Facilitated diffusion is typically much faster than simple diffusion at low solute concentrations because you have a direct route.
Right.
However, because you only have a finite number of gates, the carrier proteins, if the solute concentration outside gets too high, those gates become saturated.
The rate of transport just flatlines.
It does, like
Yeah.
traffic backed up on a bridge during rush hour. Simple diffusion doesn't saturate because the entire membrane is available, but it's slow.
Okay, that brings us to water, which feels like a simple molecule, but has these profound effects through the process of osmosis.
Osmosis is just the diffusion of water, the solvent across that selectively permeable membrane. Water moves to dilute the area with the higher solute concentration.
So it's moving from where water concentration is high, meaning low salt, to where water concentration is low or high salt.
Exactly. And this simple movement is the single biggest determinant of cellular survival, which leads to the concept of tonicity.
Let's consider animal cells first. If you place a red blood cell in an isotonic solution, like a 0.9% salt saline, it's happy.
It's the ideal environment. Water movement is balanced. But put that same cell in a hypotonic solution, lower solute concentration outside water rushes in by osmosis to dilute the cell's contents.
And the cell swells and bursts. That's lysis.
Lysis. Conversely, a hypertonic solution has higher solute outside, drawing water out of the cell. The cell shrivels or crenates.
But plant cells have a different structural integrity, which changes the dynamic.
Absolutely. Plant cells have that rigid cell wall. In their natural, healthy state, they are in a hypertonic environment.
So water moves in.
Water moves in, the central vacuole expands, and the cell presses against the wall. This is called turgor pressure, and it's what gives non-woody plants their structural support. They are turgid.
And when the environment becomes hypertonic,
Water leaves, turgor pressure drops to zero, the cytoplasm shrinks away from the cell wall, and the plant wilts. Plasmolysis.
our sources used a great analogy for the ultimate stress test on these gradients, the double whammy faced by fish in different environments.
It's a fantastic way to conceptualize life-defying passive transport. A freshwater fish lives in an environment that is highly hypotonic relative to its body, so it constantly faces two problems.
Water floods in, and vital ions diffuse out.
Exactly. Water floods in, threatening to dilute their body, and those essential ions tend to diffuse out.
So their entire existence is basically dedicated to solving this water in, salt out problem.
Yes, they adapt by developing mechanisms to rapidly excrete a massive amount of dilute urine, and critically, they use active transport pumps in their gills to constantly take up ions from a very low external concentration.
They are fighting the gradient every single second.
Every second. And saltwater fish have the precise opposite battle.
Dehydration.
High osmotic potential outside means water is constantly drawn out of their gills, they risk dehydration, while excess ions tend to diffuse in.
So they have to drink water and then pump the salt out.
They do. They actively excrete huge amounts of excess ions, again, using highly specialized pumps in their gills, to match the salinity of the sea. The constant battle against the concentration gradient is truly life or death for them.
That brings us to the logistics of fighting the current. If passive transport is riding the river downstream, active transport is fighting upstream.
And this demands energy ATP because we are moving molecules against their concentration gradient, from low concentration to high concentration.
This is, non-negotiable for life.
It is, because if the cell didn't have active transport, those essential concentration gradients, the very potential energy that drives cellular function, would immediately dissipate.
So the canonical example of primary active transport where ATP powers the movement directly is the sodium-potassium pump.
The knock ATPase, yes.
We often hear the rote description. It moves 3 sodium ions out and 2 potassium ions in. But the real insight isn't just the movement, is it? It's the fact that it creates an electrical imbalance.
Absolutely. The net movement of 3 positive charges out for every 2 positive charges, it means that the inside of the cell becomes more negative relative to the outside.
It creates a fundamental electrochemical gradient.
That's the engine. It's the charge separation, the voltage, that dictates whether a nerve cell fires or a muscle contracts.
So instead of listing all 6 chemical steps, let's focus on the choreography. How does the cell ensure that elegant, precise, 3 out, 2 in sequence happens using ATP?
Well, the magic is the energy investment. The pump uses the energy released from ATP to physically pivot or change its conformation.
Okay.
When sodium binds on the inside, the ATP phosphorylation forces the protein to rotate, opening up to the outside and releasing the sodium. Then, when potassium binds, the phosphate is cleaved off, forcing the protein to snap back to its original internal conformation.
Releasing the potassium inside.
Right. It's an elegant, energy-powered physical rotation that ensures the pump is never open simultaneously to both the inside and outside, maintaining security.
And that potent electrochemical gradient then becomes the fuel for secondary active transport.
It's cellular capitalism at its finest. It's a synergistic system. Secondary transport capitalizes on the massive potential energy stored in that primary gradient, that huge pileup of high extracellular sodium.
So you use that sodium wanting to rush back in to drag another molecule along with it.
Exactly. And based on direction, we have two types. Symporters transport two solutes in the same direction.
Like the sodium-dependent glucose cotransporter. It uses the inward rush of sodium to pull glucose into the cell.
Right. And then you have antiporters, which trade. They transport two solutes in opposite directions.
Like the sodium-calcium antiporter, which uses the inward energy of 3 sodium ions to push 1 calcium ion out.
That's the one.
Before we move to disease, we have to talk about the conceptual surprise our sources uncovered regarding channel efficiency. Specifically, how optimal flow is achieved not just through attraction, but through repulsion.
This is truly the theoretical edge. We assume efficient transport relies on perfectly designed, attractive binding sites. And that's true, but only when external concentrations are small.
Attraction helps the molecule find the channel and move through.
Yes. But when concentrations are high, when molecules are crowding the entrance, the sources show that attraction actually becomes counterproductive.
The queue is just too long.
Exactly. When the queue is massive, attraction slows down the flow because the molecule lingers too long at the binding site, creating a bottleneck. The most optimal high-efficiency transport is surprisingly achieved using repulsive binding sites.
A calculated shove. It sounds like the channel is acting as a bouncer saying, "You're in, now move along quickly."
That's precisely the concept. Repulsion shortens the mean residence time of the particle in the channel. It balances the time needed to enter versus the time needed to exit the site.
So if the binding site is attractive, the particle spends too much time there, holding up the line.
Exactly. A repulsive site ensures the particle is kept moving, maximizing flux when external concentrations are saturating the system. And furthermore, the theory shows that this asymmetry matters.
Where you put the shove.
Right. Placing that repulsive site right at the entrance is mathematically more efficient than placing it further inside.
That is fantastic. The ultimate efficiency is found not in pampering the molecule, but in strategically repelling it to maximize turnover.
Incredible, isn't it?
Seeing this level of sophisticated control and transport makes it very clear why defects lead directly to major disease. The first, and perhaps most famous, is cystic fibrosis.
Cystic fibrosis, or CF, is a prime example of channel failure. It's an inherited disorder caused by a mutation in the CFTR gene, which codes for an ATP-gated chloride channel.
And the impaired function means chloride can't move out of the epithelial cells.
Right, can't move into the mucosal layer. And the symptom cascade begins because water follows salt.
Since chloride is trapped inside the cell, water fails to follow it to hydrate the mucus.
And this results in incredibly dehydrated mucosal surfaces, leading to that thick, sticky mucus that defines CF, causing recurrent pulmonary infections and often pancreatic insufficiency. It's a system breakdown that starts with one faulty channel.
On the flip side of that, we have diseases where the channel is overstimulated, like cholera.
The cholera toxin is particularly devastating because it indirectly hyperstimulates that very same CFTR chloride channel in the intestinal lining.
So it drives massive amounts of chloride out of the cells and into the gut.
It does, and water floods out of the bloodstream to follow the chloride, resulting in the massive fluid loss and severe watery diarrhea characteristic of the disease.
Too much flow is just as dangerous as too little.
For sure. We also see targeted failures in the kidney, for example, Bartter syndrome.
Bartter syndrome is an autosomal recessive defect in the sodium-potassium chloride cotransporter in the kidney's loop of Henle.
Right, and because that cotransporter fails to properly reabsorb those three ions, they're excreted, and the water that should follow them is also lost.
Which is clinically relevant because it shows the mechanism of powerful loop diuretics.
Exactly. Those are drugs designed to target and inhibit those exact same channels to promote fluid excretion.
Finally, let's look at a powerful pharmaceutical intervention. cardiac glycosides, like digoxin, which is used to strengthen a failing heart.
Digoxin provides a beautiful, indirect clinical application of active transport. It acts by directly inhibiting the NAC-A ATPase pump in the heart muscle cells.
So if the pump is inhibited, sodium starts to pile up inside the cell.
Precisely. And that elevated intracellular sodium then throws off the equilibrium of an adjacent secondary transporter, the sodium-calcium antiporter.
Because the sodium gradient is now weaker, the antiporter slows down.
It does, meaning less calcium is pumped out of the cell.
So the net effect is more calcium stays inside the cell.
Correct. And since calcium is the trigger for muscle contraction, increasing intracellular calcium enhances cardiac contractility. Digoxin leverages a chain reaction of inhibition on basic transport pumps to create a life-saving positive inotropic effect.
This deep dive has shown us that the cell membrane is anything but a static barrier. We moved from the foundational fluidity maintained by cholesterol's sophisticated temperature buffering.
Through the energy-free dynamics of osmosis and the extreme adaptation required for fish survival,
And finally, into the energy-driven world of building electrochemical potential and its profound clinical implications.
the synthesis here is that the ultimate efficiency of life hinges on an incredibly intricate, highly specific balance of forces, gradients, and precisely structured proteins governing movement.
Every transport decision, whether passive or active, is a crucial step in maintaining order against chaos.
He really is.
So what does this all mean for the bigger picture?
Well, the sources showed that specialized membrane channels can be made maximally efficient not only by attracting necessary molecules, but sometimes by repelling them at key points to prevent molecular bottlenecks. If evolution has tuned the subtle forces of attraction and repulsion within channels, creating an asymmetric landscape of molecular shoves and pulls to achieve optimal flow, what complex, non-intuitive flow mechanisms might exist in other biological structures that we currently interpret as simple, random diffusion? The precise engineering landscape within every biological channel remains a frontier for understanding the ultimate efficiency of life.